All right, hey, thanks, and thanks, Colby, for taking the hardest slot of the day. So I'm coming from Ginkgo Bioworks, which is just down the street at Dry Dock, and I'm only briefly going to be discussing what Ginkgo does. I was invited to talk. We've been a long time user of Nextflow and a commercial subscriber to Tower. And the story that I kind of want to tell is a little bit about, it's sort of a mix of a personal story and a professional story with why I like Nextflow. And so it's going to be, the story arc is going to be a little bit about some of the problems that we've been trying to solve, a couple of the differences in the corporate context going from academia, which I won't really mention, to a small startup to a much bigger startup, and why Nextflow has been an effective collaborator with me to deliver results in these different environments. So the common thread that comes through all of this is going to be around building and using a unique genetic database from which we're going to mine different types of genetic parts for industrial purposes. And the core idea was born out of the basic idea, reinforced in the sequencing era, that the vast majority of interesting microbes that live out there actually haven't been cultured. Maybe interesting is the wrong word, but the deeper you sequence, the more that you find. And so the immediate industrial or exciting industrial application is that if microbial life has been the source of some of the interesting antibiotic, antifungal, and anti-cancer compounds, and you have this huge undiscovered biodiversity, wouldn't it make sense that there's biochemistry encoded in these organisms that can make new and interesting natural products for medical purposes? And so this sparked quite a bit of venture capital interest. I have up here the timeline of Gko Bioworks, my current employer, as well as a number of companies that were ultimately brought into Ginkgo via acquisition. I was one of the co-founding scientists of Lodo Therapeutics, which was subsequently bought by Zymergen, and now quite a few of our scientific and data assets live within Ginkgo. So at Lodo, the basic idea of how we were going to approach the problem of capturing uncultured microorganisms is we're just going to clone it. And my postdoc advisor, Sean Brady, basically came up with this protocol here, which was published in 2007, where just like you and your children do with bananas at the museum, you're basically going to take we basically just took soil, we bulk extracted and purified high molecular weight DNA. You can clone it, you can package it in phage, you can infect E. coli, and then you can have from a single soil, the rough equivalent, genome equivalent of a culture collection. And we could do this over and over again, and the idea would be that we would amass a unique genetic resource that we could exploit for natural product discovery. So within Lodo, we didn't have a huge amount of money, and we used targeted sequencing. When we were acquired by Zymergen, we invested in deep sequencing these various libraries. And when we got acquired by Ginkgo, those libraries were joined together to create what we've been calling the Unified Metagenomic Database, which is the aggregated genetic resources of the various companies that I mentioned. The major thing that I'd like to just point out on the right-hand side, I don't have detailed information about our database, but to the best of our knowledge, if we take every single available protein sequence and cluster it with our database, we are not nearly doubling the size, but we have basically 40% of our sequences don't show up in any of the public databases. So we think we have a unique and powerful resource. And although I'm mentioning proteins here, I think there's some extrapolation out into the DNA and RNA that might be in these data. So from a synthetic biology application, what does that do? You have this big database, what are you going to do with it? There's a couple of different classes of problems that we might want to solve. One is this natural product discovery, which I mentioned. Another obvious one would be an enzyme and protein discovery, and then using DNA or RNA parts for a number of different applications. And you can see the companies I've worked at and their interest in these areas. So for natural product discovery, just to give a flavor, the basic idea would be you would use some sort of sequencing in your library to identify a motif of interest, use it to physically recover the fragments which you've previously cloned, isolate those, piece them together, express them in a heterologous host, isolate the compound, characterize the compound. So it's a long, expensive process, but this is one of our successes. This is a 2018 paper, and this is describing a new antibiotic in the calcium-dependent antibiotic family with a new mechanism of action. So it's a tantalizing and wonderful idea, still unfortunately quite difficult to do. So one of the most useful application, this is where we've really, I think, hit the nail on the head within Ginkgo, is taking these data assets that we've been generating and the workflows, which I'll get to, for using these assets and applying them for industrial projects internal to Ginkgo. So the obvious approach for the thing that you'd want to do is the classic enzyme discovery. And in this case, someone comes to you with a project. I have this enzyme. I have this process. We need to optimize it. And one of the things you might want to do is go out into the world and find better acting natural versions of those proteins. And that's sort of the bread and butter, one of the bread and butter processes that we're doing within our team. You can see that we have a number of other potential approaches. The other big approach that's exciting and getting areas is that if you have a unique large data asset, you can potentially use it to create a foundation model, potentially using existing neural net architectures or potentially exploring your own. We're looking at both of these opportunities. Within protein engineering, I'll just flip through a couple of the ways that we're using this. So this is this idea of metagenomic sourcing, and this is using nature's pre-existing sequences to identify high-value candidates for usually an industrial process and usually a chemical transformation of interest. We are also in the team using Nextflow and other workflow tools to optimize things like selectivity between a desired product and an undesired contaminant from enzymatic transformation. And lastly, I think one of the areas that we've really excelled, and this is exciting, is just the iterative design of proteins towards a targeted goal. We've become quite good at that, and sort of these are 3 typical examples showing 10x or 80x improvements after 3 rounds of targeted mutation analysis. So in this case, if we have the initial data, which I was discussing in terms of the large metagenomic database, we are also matching that with tagged pairs of assay data, labeled data. So we have a lot of programs. The main point here is that if you have a protein optimization problem, you should come talk to us. I have to run, but Zach Kurtz here, my longtime collaborator friend, is here. And so this is the synbio landscape in which I've been in for the last 8 years. So why do I like Nextflow? Why am I here? Just to give a little bit of context, Lodo Therapeutics was only around 20 people. We were the chief cooks and bottle washers, like anything that remotely involved a computer or a cloud, we did. By the time you get up to a company that's 1,000 or 1,500 people, the specialization is different, the permission access is different, the ability to get things done is different. And one of the ways that I think that we've been managing to be effective is through our choice of data organization and the technologies, and one of those technologies is Nextflow. And so I'd like to just highlight a couple of the things that I found to be very useful. This might be useful to you in your careers, or it might resonate. So I think there's a couple of areas where Tower really seems to excel, and the first is general async batch compute. We've heard a number of people talk about that. I mean, that's basically what one needs to do in bioinformatics in a company, and there's certain properties of this that make bioinformatics less useful for other workflow systems, or maybe Powell and others have really sort of understood that this spiky compute operations with massive I/O, variable compute, and steps that don't have necessarily matching compute problems is well matched by Nextflow's operational model. Tower also handles a bunch of the sort of permissions muck. The other major idea, and we just heard in the last speaker of, well, everything's not a pathway, like everything's not a pipeline. In a company, you often have to ideate very, very quickly, and Tower or Nextflow and then Nextflow plus Tower offers a pathway to go from a little cobbled together thing to a production in an iterative step, instead of, for example, having a bioinformatic guy over here and then you throw it over a fence to your engineering team. So having one group own everything vertically is incredibly powerful. If you haven't been in a big organization, you haven't experienced that problem quite yet. You can also layer things on top of Tower, so both via API calls for things like triggering jobs upon an event, or also offering internal tooling that are custom to your like internal stakeholders. And lastly, and I think the thing that wraps all of this up, is that it fundamentally puts the domain expert in the driver's seat and the first-class citizen. So we'll now enable you or me as bioinformatics scientists to not worry about engineering details and be able to deliver high-quality reports to your end users. And so this is like maybe a schematic of what that would look like. On the left-hand side, we have our data. Here it's UMDB data. You can imagine anything. On the right-hand side, we have users, which are ourselves, other technical people, but also people in BD and potentially legal, and they don't need to know anything about what's inside that gray box. They don't really need to know anything that's on the left-hand side, but in that gray box is a system with a small engineering team, only 1 or 2 dedicated servers, a lot of open source parts, and basically it favors us. It lets us work quickly. And I think it shows, this is like some stats that we have for workflows internally. At Ginkgo, you can see that we introduced the system not that long ago. We were acquired late 2022. But you can see we were up and running pretty quickly, and we've seen a growing use both in the top in terms of the number of pipelines that are created. On the bottom left, the number of pipelines by team, so we're seeing adoption across multiple teams. And on the bottom right, these are the number of programs, so various customer programs that we're using these pipelines on. And in all cases, we see this upward trajectory. So people are using it. If you haven't used Nextflow, I'm assuming everyone did, like one of the killer features is the implicit parallelization. You can get something running on a small number, and then if you want to run it on 1,000 or 100,000 or 1,000,000, you can basically do that without having to write any other code other than your credit card number. So this is like, I think, the killer feature, and one of the, I think, addictive aspects of being a bioinformatics scientist, if you guys haven't experienced this, is turning the knob up and seeing like thousands or hundreds of thousands of CPUs just start lighting up. That's a really wonderful feeling, and it's very easy to do, maybe too easy. This is just a case study of someone who was brought on from a different company. They didn't use Nextflow. We helped onboard a pipeline. They loved it. This is their literal words. They love being able to introspect on their data. The reason that we started adopting Nextflow Tower as opposed to Nextflow and as opposed to the private version of Tower was the AWS Forge, which we used to write this stuff first manually and then via one of the templating systems, and it is not a good way to spend your life. So this was the initial driving force between getting Tower. It completely abstracts away all of the AWS or other cloud vendor interstitial tissue. You don't need to care about it. Your things will run. It's wonderful. Reports, I mean, I'm maybe not on the social media as much, but to me this is like the killer feature. Well, the second killer feature, which is being able to provide utility to the people that care about the thing that you're doing. And so before reports was announced, like basically all of these things, we had shadow versions of them, and then when they were brought on by the team, we immediately adopted them. So being able to provide a stable URL that has interactive access to your data is an amazing killer feature. The high I/O problem, I think this is the fundamental differentiating technical aspect of bioinformatics. I don't think it's solved. I'm looking forward to the Fusion system. We found Nextflow is better than other systems, but by and large, we're pretty happy with the system that we have. We're using a Lustre drive. Looking forward to Fusion. And we haven't been embracing the open source just as much, simply because a lot of the stuff started before, but we're definitely appreciative of all of the contributors out here. There are a couple of things that aren't, could definitely improve. I don't want to dwell on them, but I think we all know them, the developer tooling, the testing setup. It would be nice to have some additional features later on. I had some great conversations with some of the Nextflow team today, so we're hoping to do those. And so the take home that I have, even though we started about synbio, is basically the major value proposition in my mind of Nextflow Tower is that it is a tool that you can rely on that will make you, as a bioinformatics scientist in an organization of almost any size, be more effective in your job. And so that's kind of the take home. Thank you.